Everyone, and welcome to this audio podcast as part of another an audio podcast as part of the Labside Archetype Edition. This is an audio podcast as part of another Labside Alchemy a radio edition and the Storm Robotics production. This is an audio podcast as part of another Bostics blast. In this audio podcast, there'll be an overview in around works that originally was sort of involved in around 2003, and in 2023 that this works had a sort of revisited, and this works there have been and this works had been sort of presented at the UK KW 2023 conference, at the UK KW 2023 conference, and this works looks at observing chemokine and chemokine receptor expression in acute allograft rejection, or AAR, and looks at a retrospective on human transplant immunobiology and laboratory practice. So in around the introduction, there are now a sort of extensive data and investigations or studies on the observation of many chemokine and chemokine receptors in human renal transplantation or transplantation that show sort of an increased expression in acute allograft rejection, or AAR. The chemokine receptor CXCR3 has been established as a significant immunological component in AAR. In patients or recipients who develop AAR, circulating CXCR3 expression is significantly upregulated, and in literature, in literature in context of upregulation of CXCR3 by day 3 is identified or is evident in around by day 3 post transplantation. CXCR3 positive activated T cells are present in the cellular infiltrates in AAR. CXCR3 positive T cell infiltrates are also localized to sites of high IP10 and CXCL10 expression. Published figures or works vary or highlight from anything from 30 to 90% in context of CXCR3 in context of CXCR3 migration, CXCR3 migration and expression. In context of literature, laboratory investigations have informed that in transplantation, there are alternative pathways that promote the development of pro-trafficking phenotype of T cells in recipients that have received anti-CD25. The hypothesis in around this works was addressed by using a T cell culture system to analyze the effects of anti-CD25 on the expression of CXCR3 and other significant chemokine receptors on the surface of T cells. In around the aims and objectives, this works look to provide a retrospective overview of observing chemokine and chemokine receptor expression in AAR. in context of transplantation and transplant immunobiology. In and around the discussion, in contrast to the escalating data and analysis, data from analysis of roles of chemokine pathways in AAR, exploration of the role of unsatisfactory nature of sorry exploration of the role of chemokines in mediation of chronic rejection research is still in its infancy, primarily because of the unsatisfactory nature of laboratory models of AAR. In conclusion, around the conclusion of this and this as part of this in this as this works with this works, or part of this works, research retrospectively informs that there are direct effects of chemokines on recruitment, proliferation, and activation of vascular smooth muscle and other hemopoietic cells, in addition to mediation of leukocyte recruitment. Induction of CXCR3 and IP10 mRNA has been associated with AAR, aligned to human renal transplantation, or human transplantation and transplant, and around transplant transplant immunobiology. Some terminologies that can be used to identify similar works around this include acute allograft rejection, chemokine receptors, CD25, CXCR3, renal transplantation, and immunobiology. That draws this overview to a close. This has been an audio podcast as part of a Labside Archetype Edition. This has been an audio podcast as part of a Labside Alchemy, an audio podcast as part of a Bostix Blast, and an audio podcast as part of another Storm Lobostics production.